Dear students, in this module, knowing about the qualitative and quantitative advantages of UV visible spectroscopy to identify the charge transfer spectra in various compounds, to evaluate dissociation constants of various acids and bases, and to analyze the thermal denaturation of DNA using UV visible spectroscopy. Okay students, so let us now introduce the topic of this applications of UV visible spectroscopy. UV visible spectroscopy, as we have already taught you in the previous modules, is a technique that readily allows one to determine the concentrations of substances and therefore enables the various scientists to study the rates of reactions and determine equations for reactions from which a mechanism can be proposed. And thus you can now see how important UV visible spectroscopy becomes for scientists in their daily experiments. Now, next is the applications of UV visible spectroscopy. Students, as such, UV visible spectroscopy is used extensively in teaching, research and analytical laboratories for the quantitative analysis of all molecules that absorbs ultraviolet and visible electromagnetic radiations. The first application of such will be qualitative analysis. UV visible spectroscopy has been exploited for the structure elucidation of organic compounds, detection of their different organic functional groups present in the mixture and different organic compounds present in the mixture, and their separation by several analytical techniques such as thin layer chromatography. First, in this qualitative analysis, we would like to take up with you the structure elucidation of organic compounds. Now, UV spectroscopy is very useful in the structure elucidation of many organic compounds. The presence or absence of a particular absorption band at a particular wavelength may be regarded as an evidence for the presence or absence of a particular chromophore in the compound, and thus this is helpful in the schematic structural elucidation of various organic compounds. Next comes the determination of impurities. Now, as you know students, TLC, that is thin layer chromatography, is a very simple technique and is commonly used for the qualitative analysis of the reaction mixture. Now, there are many organic compounds which absorb UV light of various wavelengths in the UV range. Now, this can be used for the determination of impurities in the organic compounds. A small spot is kept on the TLC plate or is marked on the TLC plate, which is then allowed to run in the appropriate solvent media. By appropriate, I mean of appropriate polarity. Now, different compounds travel different distance based on their polarity, which can be visualized on the TLC plate under a UV lamp, depending on which chromophore absorbs in the UV region. Now, extra spots on the TLC plate can be observed due to the impurities in the sample, and it can be compared with the spot of the standard sample. Now you can realize how important UV spectroscopy becomes in determination of a particular organic compound. Now students, a concept of UV visible spectroscopy may be used as a detector for HPLC. HPLC is a very high-end purification technique in chemistry, and it stands for high performance liquid chromatography, formerly referred to as high pressure liquid chromatography. Now, UV detectors usually of variable wavelength, and they can be used to detect the molecules with absorption maxima above 210 nanometer by measuring the absorbance of the eluate. Now, it provides a very fast detection and separation of a reaction mixture. When a compound is eluted from the HPLC column, it absorbs the particular UV radiation according to its chromophore present in the compound at an appropriate wavelength. Now, the amount of UV radiation absorbed is directly proportional to the amount of particular compound that is passing through the column at that particular time. Now, this is a qualitative analysis of that particular compound using the HPLC technique. Now students, taking another aspect of UV visible spectroscopic applications is the quantitative analysis of various compounds. Now, the first in this would be determination of the concentration of the compound. UV absorption spectroscopy can be used for the quantitative determination of compounds which absorb UV radiation. Now, this determination is based on Beer's law, which relates the absorbance of the substance at a particular wavelength to the concentration C as follows. Here in this Beer-Lambert's law, A is equal to log 10 I naught divided by I is equal to log 1 divided by T, which then is equal to minus log T is equal to EBC. Now here students, as explained earlier also, E is the extinction coefficient, C is the concentration of the sample, B is the length of the cell that is used in the UV spectrophotometer. As we already told you that the cells used in UV spectrophotometers are also known as cuvettes. So B is the length of that cuvette. I naught is the intensity of the incident radiation which is falling on the compound under study, and I is the intensity of the transmitted radiation after the compound has actually absorbed the required wavelength which is falling on it. The use of UV spectroscopy, that is the Beer-Lambert's law, in quantitative analysis can be understood by a very simple example of the estimation of a mixture of anthracene and naphthalene. Now, as you know that both these compounds have a lot of conjugated double bonds in ringed structures, thus they absorb in the UV visible region. The UV spectrum of an ethanolic solution of anthracene shows a lambda max at 375 nanometer, whereas naphthalene does not absorb in this region. Thus, from a mixture of anthracene and naphthalene, we can calculate the amount of anthracene by using Lambert-Beer's law as discussed above by me. We can measure the absorption of the mixture at 375 nanometers wavelength, and thus by using the equation stated above, that is A is equal to EBC, we can calculate the concentration of pure anthracene and hence the proportion of anthracene in the mixture. Now, UV visible spectroscopy has been used extensively in industrial analytical organic chemistry. The percentage of vitamin A and vitamin A2 in natural fats or oils can be estimated by measurement of the intensity of peaks at 325 nanometer and at 351 nanometer respectively. Thus, we can say that by comparing the intensities of the solution of these two vitamins with the intensities of the known solution of these two enables us to calculate the concentration of vitamin A and vitamin A2 separately. As you can see here in the structure, they both are conjugated double bonded structures and their intensities can be used to calculate their concentrations. Now, a similar procedure may be used for the estimation of ergosterol in fats, anthracene in benzene, carbon disulfide in carbon tetrachloride, and chlorophyll in plant material, etc. Now, you can slowly realize how important UV visible spectroscopy is in daily life as well as to experimental scientists as well as in the industry. Second comes the chemical kinetics, that is a study of chemical reactions using UV visible spectroscopy. UV visible spectroscopy can be used to study reaction rates. Now, how do we do that? Let's discuss this in detail. Spectrometric rate determination involves the measurement of absorption of either the reactants or products at a fixed wavelength. When a reaction occurs, if a reactant or a reagent or the product of the reaction absorbs radiation at a particular wavelength, the spectrophotometer can be set to measure the absorption at that wavelength and as a function of time, and hence the rate of reaction can be measured. Now, to explain this, let us now take an example for a better overview of the topic, which shows the removal of a proton from nitroethane with the help of hydroxide ion, that is in basic conditions. Here only the anion of nitroethane absorbs in the UV region at 240 nanometers, while other reactants or products do not show any significant absorbance at this wavelength. Now, in order to measure the rate at which hydroxide ion removes a proton from nitroethane, or you can say the rate at which nitroethane anion is formed, they are one and the same thing. The UV spectrophotometer is adjusted so that it measures the absorbance at 240 nanometer as a function of time. Now, nitroethane is taken in a cuvette containing a basic solution, as I have already told you, and the rate of reaction is determined by monitoring the increase in absorbance at 240 nanometer as the nitroethane anion keeps on forming. Now, this is more clearly measured or seen by you in this figure as you see the absorbance with the function of time as plotted in this graph. The most widely used application of spectrophotometric rate measurements is for the study of enzymes. Now, what are enzymes? Enzymes are proteins that are present in the living tissue. Now, enzymes cannot be measured directly. Why? Because their action is being the catalyzing of the various reactions in the body. But their catalytic properties allow their estimation from the speed of the reactions which they catalyze in the body. Now, enzymes have many uses as reagents or as labels that can be attached to the other molecules to permit their indirect detection and measurement. Now, the widest used in the field of clinical diagnosis is as an indicator of tissue damage. When cells are damaged by disease, enzymes leak into the bloodstream and the amount present indicates the severity of the tissue damage. Now, indirectly we can measure the amount of enzyme present using UV visible spectroscopy. Now, the relative proportions of different enzymes in the body can be used to diagnose diseases, say of the liver, pancreas or other organs which otherwise exhibit similar symptoms. Now, not only in industry or to experimental scientists, now you can appreciate that UV visible spectrophotometry is very useful even for the doctors and in clinical research. The most common reaction used in these clinical assays, as explained above, is the reduction of nicotinamide adenine dinucleotide, or the short form is NAD, to NADH2. Now, the spectra of NAD and NADH2, as you can see, now it is clear from the figure that if the absorbance is measured at 340 nanometers, the readings will increase as the reaction progresses towards the formation of NADH2. Now, similarly, considering another example, the enzyme lactate dehydrogenase catalyzes the reduction of pyruvate by NADH to form lactate. Here, NADH is the only species in the reaction mixture which absorbs light at 340 nanometers. Hence, like the earlier example, by measuring the decrease in absorbance at 340 nanometer, the rate of reaction can be determined and its kinetics can be studied. This is clearly shown in the figure. As you can see, the absorbance is plotted as a function of time. Now, we come to the third aspect, which is the dissociation of constants of acids and bases. Now, as you might have studied in the past classes, the pKa of a compound can be determined by the UV visible spectroscopy if either the acidic form or the basic form of the compound absorbs UV or visible light. Now, taking an example, the phenoxide ion has a lambda max at 287 nanometers. As it has increased conjugation, if the absorbance at 287 nanometers is determined as a function of pH, the pKa of phenol can be calculated by using Henderson-Hasselbalch equation. Now students, if you remember, Henderson-Hasselbalch equation is pH is equal to pKa plus log concentration of the anion divided by the concentration of the acid. Now students, by plotting a graph between the absorbance versus the wavelength at different pH values, the ratio of the concentration of anion compared to the acid or divided by the acid can be determined, and hence the pKa value of the compounds can be calculated. Now the next is the quantification and thermal denaturation of DNA. Now students, this is a very specific example and it determines the concentration of DNA in single-stranded and double-stranded form, and it shows how important UV visible spectroscopy is in various fields. Now UV visible spectroscopy can be used to estimate the nucleotide composition of DNA. The two strands of DNA are held together by both AT base pairs and GC base pairs. Now when DNA is heated, the double-stranded DNA breaks down. Single-stranded DNA, which results, has a greater molar absorptivity at 260 nanometers than double-stranded DNA. The melting temperature, also denoted as Tm of DNA, is the midpoint of absorbance versus temperature curve as shown in the figure. Now clearly, for a double-stranded DNA, Tm increases with increasing number of GC base pairs because they are held together by 3 hydrogen bonds, whereas the AT base pairs are held together by only 2 hydrogen bonds. And hence, this can be used to estimate the number of GC pairs in DNA. Now, next students, we come on to the study of charge transfer complexes. The formation of charge transfer complex occurs between molecules which, when mixed, allow the transfer of electronic charge through space from an electron rich center to an electron deficient molecule or electron deficient center, with molecular orbitals having suitable energy and symmetry to do the same. Now students, as you can see, there are many charge transfer acceptors as shown here, and as well as charge transfer donors. You can see both type of molecules in the figure. The filled pi orbitals in the donor molecule, as you can see, overlap with the depleted orbital in the acceptor molecule and generate 2 new molecular orbitals. Thus, transition between these newly formed orbitals are responsible for the new absorption bands, which are observed in the charge transfer complexes. The 2 new molecular orbitals formed and the transitions between them are clearly shown in the figure, as you can see. You can see the donor's highest occupied molecular orbital as well as the acceptor's LUMO, that is the lowest unoccupied molecular orbital, and the charge transfer complex and the transfer of electronic charge through space from the donor to the acceptor. Now the brown color of iodine in benzene or the appearance of deep blue color when tetracyanoethylene is added to a chloroform solution of aniline may be explained due to the formation of electron charge transfer complex. The structure of most charge transfer complexes can be visualized as a face-to-face association on a 1:1 donor is to acceptor basis to provide maximum overlap of pi orbitals of benzene ring. Now, this is also clearly shown in the figure, whatever examples I have taken just a while ago. Now, the lambda max of benzene is 255 nanometers, while for iodine in hexane is 500 nanometers. The charge transfer complex, that is benzene and iodine, displays an intense additional band at 290 nanometers. Similarly, in the aniline tetracyanoethylene complex, lambda max for aniline and tetracyanoethylene are 280 nanometers and 300 nanometers respectively, while the deep blue complex has a lambda max at 600 nanometers. Now you can appreciate the extent of charge transfer in these various examples cited. Or the next application of UV visible spectroscopy is the preference over two tautomeric forms. If a molecule has two tautomeric forms, then the preference of one structure over the other can be detected by the use of UV visible spectroscopy. Now, taking an example, in case of tautomeric forms of 2-hydroxypyridine and pyridone 2, the later is preferred because it is an alpha beta unsaturated ketone, and clearly the equilibrium shifts towards the right hand side, and this is clearly shown in the figure. As you can see, the preference is more towards the 2-pyridone structure because it is alpha beta unsaturated carbonyl compound. Now next is the application of UV visible spectroscopy in the determination of configuration of geometrical isomers. The cis geometrical isomer suffers distortion and is forced to be non-coplanar and thus absorbs at lower wavelength. In comparison of the trans geometrical isomer, it suffers no such distortion and thus it absorbs at a much higher wavelength. Now you can see in the figure that the trans stilbene, taking an example of trans and cis stilbene, the trans isomer absorbs at 295.5 millimu, which is higher than the cis. Now students, the next use of UV visible spectroscopy is the distinction in conjugated and non-conjugated compounds. One can easily distinguish between conjugated and non-conjugated compounds by using UV visible spectroscopy very effectively. The N to pi star band for the carbonyl group, as you can see in the structure 1, will appear at a longer wavelength as it is an alpha beta unsaturated ketone. As you can see in the structure, it is conjugated. The carbonyl is conjugated with the double bond present. However, in comparison to the absence of conjugation in compound 2, where the double bond is separated by a methylene group, thus the conjugation is broken. Thus the same band appears at a shorter wavelength. Now coming to the next use of UV visible spectroscopy is the differentiation between the equatorial and axial conformations of various structures conformations present. The N to pi star transition, which comprises of the R band, appears at a longer wavelength in alpha beta unsaturated ketones and is strongly influenced by the presence of a polar group in the gamma position. Now it has been noted that the effect of an axial substituent to displace the R band to a longer wavelength is greater as compared to that observed in its equatorial isomer. This is clearly shown in the figure, as you can see. Now coming to the other uses, summarizing the general uses of UV visible spectroscopy. UV visible spectroscopy is also used in the quality control in the development and production of dyeing reagents, inks and paints, and the analysis of intermediate dyeing reagents. In environmental and agriculture fields, the quantification of organic materials and heavy metals in fresh water can be carried out using UV visible spectroscopy. A special type of UV watermark is kept on many sensitive documents such as credit cards, driving licenses, passports to prevent forgery. The watermark can be only seen in the UV light. The optical whiteners absorb ultraviolet light and re-emit in the visible range. This feature is used in washing powders. Optical whiteners are also added to many toothpaste and detergent powders. Now students, not only the use of UV spectroscopy, here we also appreciate the chemistry being used in every phase and every walk of life. UV visible spectroscopy forming a very important part of chemistry. Now, summarizing this module, students, let us now see what we have learned in this module. As you know, we have already come to appreciate a lot of uses of the UV visible spectroscopy. How the substituent affect the absorption pattern in benzene and other mono or di-substituted benzenes. Different applications of UV spectroscopy were taught to you to qualitatively and quantitatively determine the concentrations of various compounds present in a mixture or solely present as a compound. Studying the thermal denaturation of DNA using quantitative technique of UV visible spectroscopy. Now students, here we have definitely finished the UV visible spectroscopy, but I think it is much more beyond. The uses are also can be extended much more beyond simple experiments like I have stated here. And as science progresses, the UV visible use and its applications will definitely progress and make the science more helpful in detection of unknown organic compounds.